This meeting is now being recorded.
Okay, good afternoon, everyone. Welcome to the Office of Information Management and Analysis Brown Bag webinar series. We'd like to remind you that OIMA Brown Bag webinar will be held every month. The next webinar will be on Thursday, July 28th. So this webinar will be strictly 30-minute event for both presentation and questions and answers. Today's seminar is about calculating California Stream Condition Index, CSCI. The speaker is Calvin Young. Calvin is an environmental scientist in the Surface Water Ambient Monitoring Program Unit at the State Water Resources Control Board. He is the bioassessment coordinator and is the point contact for maintaining the online California Stream Condition Index, SCCI course map. With this brief introduction, I will pass the phone line to Calvin to start his presentation.
Thank you, Deweet. Let me just click on to my next slide. So, thank you for the introduction. I'll just be presenting an overview of how to calculate the CSCI, or California Stream Condition Index, using GIS, or Geographic Information System, and R, which is a statistical programming language. So just to give you a brief overview of what the CSCI is, it's a new statewide tool that translates complex data about individual benthic macroinvertebrates, or BMIs, living in a stream into an overall measure of stream health. Calculating the CSCI is a two-step process. The first step is to use GIS to calculate environmental predictors, which is things like watershed area and rainfall. And then the second step is to use those predictors along with your BMI taxonomy data to calculate the CSCI in R. So here's a graphic about what the CSCI score is. This is from the CSCI fact sheet, which is on our Swamp Bioassessment website. In short, or in basic terms, it's basically the CSCI score is a measure of how well a site's observed condition matches its predicted or expected condition. So the expected conditions, so things like geology, climate, and watershed size, that's calculated using GIS, and that's from statistical models. And then the field crews go out to the site, collect the BMI information, and then they deliver those samples to a BMI lab, who then identifies the bugs. And then you compare what bugs you got to what you expected to see there. And then here's a list of the different components of the CSCI. The section that says measures of ecological traits, so percent EPT and percent intolerant species to pollution, that's part of the multi-metric index, or MMI. And then the section here says species for taxonomic completeness, that's the observed to expected index. So both of those indices put together is the CSCI. For statewide assessment of using the CSCI, there's 4 categories. So a score that's greater than or equal to 0.92, that site is likely to have intact biological condition. Scores that are less than or equal to 0.62, those sites are very likely to have altered biological condition. And then there's 2 intermediate categories for the scores that fall in between. The CSCI was calibrated during development so that the mean score of reference sites is 1. So what that means is that scores that approach 0 indicate great departure from reference condition and indicates degradation of biological condition. And then scores that are closer to 1 are reference. For more information about the CSCI tool itself, you can go to this document here on the left. It's the new SWAMP technical memo that summarizes the development and key features of the CSCI. For more information about calculating the CSCI, you can go to this document here on the right. It's the interim instructions. It basically will go over the steps that I'll go over today in greater detail. And both documents are available on this SWAMP Bioassessment website link. Also on that website is the scientific papers that are published and peer-reviewed for the CSCI and reference. Oh, and there's also an online interactive map also on the Bioassessment webpage. It's basically displaying all the sites that have been scored with the CSCI, and they're all color-coded based on which category those scores fell into. So you can click on each site or whatever site you're interested in, and it'll pop up the score, the name of the station, and the station code. I'm also currently in the process of updating the map with more sampling locations and more recent scores with help from Stephanie, who's in our GIS unit here at the water board. So if you want to calculate the CSCI, here's the list of software you'll need to download to calculate it and delineate your watershed and make sure you have access to the software. So, and then you can always use more recent versions. All right. So first step you want to do is add your site to ArcMap. And so for this example, for my review, I'm going to use a swamp station, which is called Dry Creek. It's about 0.9 miles above Cox Creek, which is in Yuba County. So here's more information about the station. So here's the station code, which is just a unique identifier of the station. It was sampled by our perennial streams assessment program, and then the latitude and longitude. So when you plot onto ArcMap, you want to make sure that you plot your site with the NHD flow lines. And so these flow lines are these blue lines here that represent each of the creeks and streams. NHD stands for the National Hydrography data set, which can be downloaded from the USGS website. And once you load onto ArcMap, what you want to do is snap your site to the flow line. And so what snapping means is that you move the point onto the line that's representing the stream that the station's supposed to represent. And the reason why you want to do that is that snapping improves the catchment delineation process using the NHD plus basin delineator, which is on the next slide I'll show you. And I guess also to keep in mind, when snapping, you want to make sure that you only move the site up to 300 meters or less, because then that could mean that maybe you're using the wrong station or using the wrong lat longs. So just to kind of show you, there's no arrow, but say that the point happens to be a little further on top of the Dry Creek line, which is kind of, I'm trying to show with my box. What you would do in snapping is you would create an edit session, you would click on the point and move it onto the Dry Creek flow line. So that's what snapping in a nutshell is. So once your site is actually on Dry Creek, next step is to delineate the watershed. So on the left side is a screenshot of what the NHD plus basin delineator is. You can download this software free from the Horizon Systems website, which is the group that created this software, and they created it for the US EPA. And basically what this tool does is, so it takes your text file that has your station coordinates, your latitude, longitude, and then turns it into a shapefile that you can then add to ArcMap. So this is just showing that you navigate to where your text file is on the top box, and then you can upload it, and you choose where to save it, and you click Analyze, and then once the process has completed, it will give you this message that says that the basin delineator successfully created your shapefile, and you'll have the files in your folder that you selected. And for this particular site, it took the basin delineator about 2 minutes to delineate. So you could do batches of sites, but just realize that the more sites you do in one batch, the longer that it takes the basin delineator to create those watersheds. So once you get your shapefile output and add it to ArcMap, this is what it will look like for this particular site. So you'll get a watershed that's delineated. Just to show you again what it looks like first, so this is how it looked at to begin with, and then this is what you'll have. And so I guess just to point out, so the goal of having these delineated catchments is to have, you want to identify the portion of the landscape that contributes runoff to a stream under natural conditions. So here's our site here. You want to make sure you capture all the streams that are connected to it within this catchment. So once you've added to ArcMap, what you want to do now is check it, do quality control checks to make sure that the right watershed was delineated. So in order to do that, what I did was I made the shapefile transparent, and then I added a topographic base map underneath just to make sure that all the streams that are supposed to be within this catchment are included in here. So these are all the different flow lines in here, and that's what you want to check to make sure that it was correctly delineated. So, for example, right now these two streams at the bottom right corner that my box is going in and out. So say that that stream was actually connected, and the basin delineator cut that in half. What you want to do is in ArcMap create an edit session and edit the vertices or edit the border so it goes around and includes that stream that was connected. But in this case, it is corrected. They're not connected, so the watershed didn't include that part of the stream. So after doing your quality control checks, the watershed looks correct and accurate. Now you're ready to calculate the environmental metrics using the CSCI metrics ARC toolbox. And what that is I'm referring to is this toolbox up here in the upper left part of the page. And each of these blue scripts are Python scripts. And what it does is after you run all of those scripts, you'll get all these different predictors that you'll get as an output in a CSV file. And for our station, this was the area of the catchment, the average temperature from 2000 to 2009, the average precipitation, average soil bulk density. These different predictors are in a CSV file. And this table down here is a table from the interim instructions that explain what each field means. So these numbers were calculated using this ARC toolbox, which then was using models to come up with these numbers. Now that you've calculated your environmental predictors, the next step is to basically download and consolidate all of your BMI taxonomy data. And so if you're using SWAMP data, then you can use this tool, which is a screenshot of the SWAMP reporting module. This will allow you to download all of your BMI information. But for all other projects outside of SWAMP, you can use CDIN or the California Environmental Data Exchange Network, which is online. You can use that to download all other BMI data outside of SWAMP. But you could also use CDIN to download SWAMP data too. So what you do is you go to cdin.org, you go to Find Data, you click on Benthic, and then you can click on the certain station that you want to download your BMI data from. So In either case, you just need to format your data into the right format so you can input it into R, and the CSCI package that's in R can run the table. So here's the table for our site that I'm going over. So you'll have your station code, your sample ID, which is a concatenation of your station code, sample date, collection method code, and field replicate number. And then you have your final ID, which is all the taxonomy names, distinct, by-stage code, and then bar result. And for data that's from seeding or outside of SWAMP, the one thing I want to point out is that for your final ID list, you want to make sure that those taxonomic names match the values that are in the SWAMP organism lookup list, which is this link here. If the names don't match or have a lot of mismatches, then the CSCI package may fail to run. So once you've downloaded all of your BMI data and you've formatted it, next thing to do is to make sure you have the R software. So here's a screenshot of the R software that I have on my computer. It's version 3.1.2, but you can always download the more recent version. And to download the software, you just go to rproject.org. So just to show you what it means to just have all your data compiled and formatted. So here's all the environmental predictors data that I have in this table up here, and I called it final metrics consolidated. You can call it each of these files whatever you want, but it's just to make sure that you remember what it's called. So when you do run R, you do have to type in the code that says use this file. So it'll be very, you have to make sure it matches. And then this table down here is just the taxonomy data that I formatted for our site for the date that it was sampled. And I just called it Dry Creek taxonomy formatted data. So once you've installed R, the next thing you want to do is install the CSCI package and then load it. And so that's this screen here. So here's all the different packages that I have in my version of R, but when you want to load packages, you just go to the option that says load package, and then you'll just click on CSCI and then push OK. And then what happens is in R, it'll give you all these different messages saying that it was all installed, and it'll all be in blue text. And so the part that you actually have to type in to calculate the CSCI is all this text that's in red. And so I circled the file names, which is this slide here, just to show you when you need to make sure you use the right file name so you can, R knows which file to pull in. So what you'll do is these first two lines of code that you type in, it's basically just telling R, so here's my bugs data, and it's called, for my case, Dry Creek taxonomy formatted data. And then my stations data, or my environmental predictors, are the metrics consolidated file. Then you type in the code, report CSCI, and you reference the bugs and the stations files that you saved. And then you'll tell R to write a CSV file called core, which will basically just give you the CSCI score and its different components. So when you do get your CSCI score, you get another CSV file with a table that shows all the different metrics that were calculated, including the CSCI. So for our site, it was, score we got was 1.05. And that's obviously a really good site that's in the likely intact biological condition category. And just to make sure I stated this, so scores that are above 1 can be interpreted to indicate greater taxonomic richness and more complex ecological function than predicted for a site given its natural environmental setting. So it exceeded expectations. And then two of the indices I wanted to point out. So here's this O over E index score. So the O over E measures taxonomic completeness, and so in our case, it was 1.2, which means that the observed taxa was greater than what was expected. So that's that number. And then the other index I wanted to point out is this MMI, or multi-metric index number. So the MMI measures ecological structure and function, and so in our case, this was 0.9. And so that means that the aggregated BMI attributes, so things like the number of shredders, how many percent intolerant species to pollution, those different attributes aggregate all together, go into that MMI score. So the 0.9 means that it was closer to reference. So that's the entire CSCI process in a very short presentation. It does take a lot longer than that to do, but hopefully this is just a good overview to get you interested into using the tool. Again, all the instructions are online on our Swamp Bioassessment website, and I am available to help if you want to do this on your own. Here's my contact information if you'd rather ask me questions later, or if you have a question that comes up later. Thank you.
Okay. Thank you very much, Calvin. Now people can ask. We'll unmute everybody. So go ahead and unmute them.
All guests have been unmuted.
People on the phone can ask now if you have questions.
Okay.
Yeah, I got a question on this one real quick. So CSCI values that are greater than 1, is there ever a readjustment to, now that you've seen that there's greater species richness, to bring it back down to like a 1 level?
No, there hasn't been a process to do that, and I would have to talk to our scientist lead to see if that would be done. That's what they plan on doing, but I don't think so. I guess just to add to that too is that there's not a lot of sites that do that do go over 1. So this happens to be just one site that I decided to choose. And I hope the screenshots help too, because visually. But I finished before the 30-minute mark.
I have a question for you.
Oh, sure.
With the expected taxa, does it take into account intermittent streams versus perennial streams for a region?
Right now, it's only for weightable perennial streams. I am not sure. They are expanding into non-perennial streams and using this, and it seems that the method is actually working, but
using the already existing method for
yeah.
intermittent streams? Okay.
Yeah.
Cool. Thanks.
Yep.
Any more questions from here? Or?
Yeah, for the CSCI values, are those posted on CNN?
So no, there's no CSCI value stored in any database for now, but Swamp and CNN is working to store those values. Right now, all the values, though, are stored onto that CSCI scores map, interactive map that I showed earlier. And then you can there's also a link to download all the scores into an Excel spreadsheet. So you can look at all the scores in a map, or download all the information in an Excel spreadsheet. So it's kind of like the official, unofficial list for now. Yeah, so feel free to contact me later if another question comes up.
Yeah, and the recording will be online, so we'll see you hopefully next month, July 28th. The next webinar is at that time. Thank you all.